Okay, this is part one of a series of lectures called Mitochondrial Insights. I'm going to talk about what mitochondria are, what mitochondria do. We're going to begin with the basics, but this is going to get to very advanced, interesting stuff. I'm just going to stick to things that are clinically relevant, useful to help you improve your health, and that are interesting. So first of all, we need to know the anatomy of a mitochondria. First, their size is about 1 micron in length. They have an outer mitochondrial membrane, abbreviated OMM. They have an inner mitochondrial membrane here in green, and that's abbreviated IMM for inner mitochondrial membrane. There's a space between the outer and the inner membranes called the intramembranous space. Okay, so intra means like in between in this case. Anyways, you could actually call it the intermembranous space. We're going to call it the intramembranous space, IMS. Okay, the center of it, which is yellow, that is the mitochondrial matrix. Okay, you need to know these terms. These are the basic anatomy terms of a mitochondria. And what's interesting is people think there's different theories, but one of the main theories is that originally an anaerobic bacteria, I'm going to talk about this more later, but the theory is that an anaerobic bacteria somehow swallowed, so to speak, a mitochondria, which was an aerobic bacteria originally, is the thought. I'm going to talk about the controversy with regard to that in a subsequent lecture. But that's the theory. And that what happened was the swallowed mitochondria then made energy for the anaerobic bacteria that had swallowed it. And the theory is that this, having originated supposedly from an aerobic bacteria, that this would be like its plasma membrane, its inner mitochondrial membrane, and its outer mitochondrial membrane would be the equivalent of its cell wall, and the intramembranous space would be the equivalent in a bacteria of a periplasmic space, the space between the plasma membrane and the cell wall. But now that it's inside of eukaryotic cells, human cells, we call this the outer membrane and the inner membrane. I'll go into all that stuff in more detail and why it actually is interesting, but just giving you the analogy to a bacteria. And bacteria are about that size too, in general, about 1 micron. Okay, here is the way glucose is brought into a cell. This is a human cell, a eukaryotic cell. EU means true, cardio means nut or nucleus. So eukaryote means true nucleus, and that's what human cells are. We are eukaryotes, multicellular eukaryotes. Okay, so glucose is brought into a cell across the plasma membrane of a human cell. There's an enzyme hexokinase. The glucose is phosphorylated, putting a big bulky molecule on it with a charge so it can't exit out the plasma membrane. It'll then undergo a metabolic pathway called glycolysis. It'll be converted to pyruvate, and then the pyruvate is sent into the mitochondrial matrix, where it undergoes a biochemical pathway called Krebs cycle. Krebs is named after Hans Krebs, who was a German biochemist researcher who worked with Otto Warburg. And by the way, the Ebden-Meyerhof pathway is the other name for glycolysis, and those were also biochemists who worked with Otto Warburg. And then Otto Warburg is the one who figured out the Warburg effect for the inner mitochondrial membrane. So the incredible reality of this is that the German biochemists figured this all out in more detail. There's obviously more to it than that, but they did extraordinary work for figuring out the metabolic pathways of glucose through glycolysis, through Krebs cycle, and what's happening in the mitochondria, inner mitochondrial membrane. And of course, we're not going to get into it right now, but Peter Mitchell in England is the genius who figured out the mechanism of ATP production across the inner mitochondrial membrane. But anyways, it is on the inner mitochondrial membrane that electron transport itself occurs, and then the process of oxidative phosphorylation. So electron transport means to hand off electrons down a chain of protein complexes. Oxphos means to use oxygen to add a phosphate to ADP, adenosine diphosphate, to make ATP, adenosine triphosphate. Okay. Okay, now here's another diagram of what happens in a cell for the production of energy. Glucose goes through glycolysis, and that only makes 2 ATPs, which is hardly anything. This is anaerobic metabolism, done without oxygen. The pyruvate, if there's no oxygen available, would be converted to lactate, and that would be anaerobic metabolism is called fermentation. Okay, so fermentation is metabolism of glucose without oxygen. When oxygen is present, though, it is preferable to use that to make energy because you get far more energy from glucose when you can run it through oxidative phosphorylation in the mitochondria. You get about 18 times as much ATP. So the pyruvate, again, goes to the mitochondrial matrix. It ends up as acetyl-CoA, runs through Krebs cycle, and then these hydrogens are extracted from the molecule, and that is used to make ATP. The way it works is you'll see CO2 being pulled off, and CO2 is really like a carboxylic acid that's been deprotonated, like a COOH with the H taken off of it. And the H is sort of the money, okay? And when I say money, what I mean by that is we're going to show in a moment that the mitochondria is like a nuclear power plant that chops the hydrogen in half, so to speak, and splits it. It splits the atom. This is like miraculous, okay? That's why when somebody tells me like they're an atheist and that's an intelligent position, I'm like, yeah, right, you stupid idiot. You don't know anything about chemistry. Anybody who knows anything about chemistry can see that this is a miracle, okay? This tiny little thing inside your cell splits the hydrogen atom into its protons from the nucleus and its electrons. These protons will be pumped into the intramembranous space, and these electrons will be passed down the carriers on the intermitochondrial membrane. Okay, and it had been said by one evolutionary biologist that in the creation of the animals of the world, God seemed to have an inordinate fondness of beetles, because there's so many different types of beetles. Okay, and it has been said by other biochemists, it seems in the creation of human biochemistry, that God had an inordinate fondness of carboxylic acids. And that's why I'm reminding you, CO2 is like a deprotonated carboxylic acid. That will become important to remember. So CO2 is like a deprotonated, and the proton is, the hydrogen, deprotonated carboxylic acid, okay? And that's what metabolism does of glucose. It basically rips off CO2, rips off the carboxylic acid, and then grabs the hydrogen, splits it in half, splits the atom, and then uses this for energy production. The CO2, we exhale that when we breathe. It's just a waste product. So CO2 is a waste product. In Krebs cycle, you can see CO2s are ripped off of there, and hydrogens are extracted as well to be used for energy production. So this is happening in the mitochondrial matrix. Glycolysis in the cytoplasm, in the cytosol. So just a quick reminder of the anatomy of the mitochondria. Outer mitochondrial membrane, OMM, like a bacterial cell wall. Intramembranous space in between the outer membrane and the inner mitochondrial membrane. This is the equivalent of the periplasmic space for a bacteria. Here is the inner mitochondrial membrane, IMM, and then in the center of the mitochondria, the yellow area, that is the mitochondrial matrix. So green is inner membrane, the white is intramembranous space, and then the black line here is the outer mitochondrial membrane. Okay. All right, this is just Krebs cycle in a little more detail. Krebs cycle is also called a tricarboxylic acid cycle because the citrate or citric acid is a tricarboxylic. Tri as in three, carboxylic is it has 3 carboxylic acids. So this would be COOH, and here it's drawn as deprotonated. When a carboxylic acid is protonated, you include the ic in its name. So this would be called, if these were all protonated, had hydrogen on there, this would be called citric acid. So sometimes you'll hear it called the citric acid cycle. Because there's 3 carboxylic acids, it's also called the tricarboxylic acid cycle. Because Krebs discovered it, it's called the Krebs cycle. Most common name given to it is Krebs cycle. And Krebs cycle is in the center of all human metabolism. It is glycolysis and Krebs cycle are the 2 most important biochemical cycles in the whole human body, okay? And this is a key point. There's 3 carboxylic acids on here, and then progressively you're going to rip these things off, okay? This third one here comes from the acetyl-CoA, from the individual pyruvate units sent into the into the mitochondrial matrix. Okay, the two carbons right here get added on to oxaloacetate to keep this cycle going in a circle, and then you end up with a tricarboxylic acid. Okay, this right here is just a little mnemonic. Okay, when I was a medical student, I had the highest biochemistry score in the whole United States, and this is how I did it. I just made a bunch of mnemonics to remember everything. So TIA is like the word for ant in Spanish, and T for tricarboxylic acid, I for the next intermediate in the cycle, isocitrate. A is for alpha, alpha ketoglutarate, okay? And then SS is for, it reminds me of McDougall's book, Starch Solution, and the first S is for succinyl-CoA. And just like the word starch is longer than the abbreviation SOLN, just remember the longer one comes first. Succinyl-CoA, and then it goes to succinate. We're going to come back to succinate a lot. Succinate is a super important molecule in Krebs cycle, super important. It interacts with a whole bunch of other pathways. We're going to come back to that. Okay. Succinate under normal conditions with oxygen available gets converted to fumarate. And I remember that like an FM radio, fumarate, because it goes from F for fumarate, then goes to M for malate. Okay, and then you turn the radio on, that's oxaloacetate. And then, so you don't need to know all this, but I'm just giving you a sense how this is comprehensible. So all you need to know is that Krebs cycle runs in the mitochondrial matrix, and you need to know that succinate is super important. There are side pathways that can come off of all these things. Krebs cycle normally in humans runs clockwise like this, and it's used to make energy. That is the normal key thing to know. But I'm also letting you know, under special situations like cancer, it can run backwards. Also, things can come and add in. You can use glutamate and glutamine to make alpha ketoglutarate. That's why you can feed cancer through the back door with glutamine. It's like the most common amino acid, and that's why I think paleo keto diets for cancer are a big mistake. Okay, because you can run all your metabolism off glutamine, so you're not going to win that game. But anyways, this is how you would remember. So you just say to yourself, Tia, my aunt, with the Starch Solution book in her hand, turns the radio on, and then I can remember everything. Tricarboxylic acid, isocitrate, alpha ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate. Okay, and then your cycle begins again, for what that's worth, if you wanted to remember it. You don't need to, but I just want you to see how this stuff is actually quite comprehensible. Okay, so here's what's happening on the inner mitochondrial membrane. Again, here's the inner mitochondrial membrane, IMM. Here's the outer mitochondrial membrane, OMM. And now the electrons are going to be passed down from complex number 1, and it's going to take this hydrogen off NADH. We got the NADH from Krebs cycle. And then the proton, the H+, the nucleus of the hydrogen, is going to be pumped into the intramembranous space. Then the electron, when we split this atom, this hydrogen, into its proton and its electron, the electron will be handed off through a series of carriers, okay? All of these protein complexes will carry the electrons, and it's kind of like a snowball rolling down the hill. And the most powerful grabber of electrons is oxygen. Oxygen has the second highest electronegativity out of any element in the periodic table of elements. And what electronegativity means is it really, really, really wants to grab that electron. It wants to steal that electron. Oxygen is a very aggressive molecule, okay? It wants the effing electron, and it's going to get it, okay? That's important, and that gives oxygen a lot of power, and it enables it to just grab that electron, and that's important because it makes this whole process run. Once oxygen grabs that electron, hydrogens are added to it, and it's converted into water, a harmless substance. You know, about 60% of the human body is made out of water. So it's a magnificent thing, okay? This is a nuclear power plant, an electricity generation plant. This is about how 90% of energy is made in humans, okay? So that's extraordinary how this all works. And again, it's a sign of the design of God. It's so brilliant and genius, you know. You could tell the greatest biochemist in all the world, get all of them together, all of them, every single one that exists, and tell them to make a machine, a nuclear power plant, you know, so small you can't even see it by itself, you know, barely with an electron microscope. they would laugh. They say, that's ridiculous. There's no way we could do that. Yet, atheist idiots think, oh, it just spontaneously designed itself. No, this is genius at a level that is beyond the genius level of any human on Earth. Okay, it's magnificent. So anyways, what ends up happening is you've pumped all these protons into the intramembranous space, and now you've built up. It's kind of like compressed air. You know how like you have compressed air and then you connect it to your car tire and the air comes rushing out to fill up your car tire? Well, that's a little bit like what goes on here. You could take the air and compressed air, use it to fill up a balloon. That hydrogens are very concentrated in intramembranous space, and then they are let to come back down into the matrix through what is called ATP synthase. It's also often called complex 5. And when the protons come through, they spin ATP synthase like a revolving door when you enter a big hotel or some other big building. And the spinning is also kind of like a mill wheel for energy production. Anyways, the spinning of ATP synthase then is used to couple adding a phosphate to ADP. Adenosine diphosphate, as in having 2 phosphates, becomes adenosine triphosphate, as in having 3 phosphates. Okay? And this ATP is the energy of life. This is the equivalent of a $20 bill. It is the cash, the money, the currency, the energy that gets work done in a cell. So you need to make a lot of ATP, and this is how you do it. Okay? And that's how it happens normally. Like a fireman bucket brigade or a snowball rolling downhill, these electrons are passed from carrier to carrier to carrier until they get to complex 4, and then they're handed off to oxygen to make water, a safe waste product. Okay? The proton's pumped in here to build up a giant gradient. This gradient's like negative 160 millivolts. It's an incredible gradient. It's the highest gradient in the human body. Okay? If you look at a regular neuron gradient, that's about negative 65 millivolts. This is 160. It's a giant effing gradient. It's incredible. Okay? That is harvested by allowing a hydrogen to come back into the matrix, and the spinning of ATP synthase is coupled to adding a phosphate to ADP to make ATP. That's how it works. Now, I do want to show you another potential problem. If this gradient gets too high, and even under absolutely totally normal circumstances, occasionally coenzyme Q will drop an electron down onto oxygen. The oxygen will be sitting around in the mitochondrial matrix, and it'll drop an electron down onto oxygen. When that happens, it'll form a molecule with the oxygen called superoxide, which is a free radical, unpaired electron in outer orbital with a negative charge. And the mitochondria is very good at neutralizing that. It has an enzyme called superoxide dismutase, and that's the beginning of, it'll just neutralize the free radical, and what you're going to end up with is water, okay, and more oxygen. So mitochondria knows how to handle that. That always happens in small amounts. We're going to talk about situations that are abnormal, pathology. Pathology means disease, where this happens too much. But just know that it always happens a little bit. And as a matter of fact, it's part of normal signaling to the cell what is happening with the inner mitochondrial membrane. Okay? Now, I just want to show you in a little more detail this idea of splitting the atom. So all these protons, we pull off 24 of them. They are used to be pumped into the inner membranous space. So that's all the red hydrogens, H+, okay? And then the green is for the electrons, and it makes an electrical current as the electrons flow down across the carriers of the inner mitochondrial membrane, okay? This complex 2 is quite unique in that it is both a member of the electron transport chain, but it is also an enzyme in Krebs cycle. It is succinate dehydrogenase, and we're going to come back to succinate dehydrogenase a lot. It's actually super, super important amongst the inner mitochondrial membrane electron carrier protein complexes, okay? But you see how this electron is passed like a fireman bucket brigade from carrier to carrier until it ends up being given to oxygen, and then the hydrogen is added back to the oxygen to make H2O water. Okay, and this is the mitochondrial matrix. Okay, and then this is the harvesting site, the complex 5, ATP synthase, where when a hydrogen proton is allowed to come back in to the mitochondrial matrix, that's coupled to converting ADP into ATP. And that's how it works. This is the IMM, inner mitochondrial membrane, intermembranous space, and above here would be the OMM, the outer mitochondrial membrane. And that's where energy comes from in life on Earth. Okay, so that's the end of part 1 of Mitochondrial Insights. I hope you found it helpful. We're going to go on in later parts to other more interesting things about mitochondria.